Good afternoon, everyone. I'm Francis Collins, Director of the NIH. It's my pleasure to welcome you to the first Wednesday afternoon lecture of 2013, with many more to come on subsequent Wednesday afternoons, and appreciate everybody who is able to gather here in the auditorium and all those many who are watching on the web, because we try to make this as accessible as possible to busy people all around this campus and in other places. We're very fortunate today to have as our presenter Dr. Barry Friedman, who's Professor of Internal Medicine and Chief of the Section of Nephrology at the Wake Forest School of Medicine in Winston-Salem, North Carolina. He has an interesting history in terms of his training, which may explain why, despite having lived south of the Mason-Dixon line for nearly 30 years, you won't detect much in terms of a perfection of the Southern accent. Barry was born in New York City, attended Brooklyn College, graduated summa cum laude, and then did his medical training at SUNY Downstate College of Medicine before departing for Virginia and then North Carolina. And since arriving in Wake Forest for a nephrology fellowship, he's risen through the ranks and became nephrology chief in 2000. He's been a prolific investigator, authored over 350 papers and book chapters. His work has been continuously funded by NIH grants from several institutions: NIDDK, NHLBI, NIAMS, NINDS. He's currently the PI on, count them, 4 NIH grants and is associate investigator on 8 others, so he has a strong work ethic. His research has focused specifically on genetic susceptibility to type 2 diabetes, as well as kidney, cardiovascular, and bone diseases, with an emphasis on the African American population. One of his contributions, which I think he will talk about later in the presentation, is the role of the apolipoprotein L1 gene and a variant in that gene in terms of predisposition to kidney disease, but also providing protection against a particular infectious disease, namely African sleeping sickness. This has been one of the most exciting stories to come out of this interface between environmental influences and genetic variation since the original description of sickle cell and its protection against malaria in the heterozygote. So I think we're in for a very interesting presentation with a focus on health disparities, a topic of enormous importance for all of us here at NIH. So please join me in welcoming to the podium Dr. Barry Friedman.
Thank you, Dr. Collins, Dr. Rogers, Dr. Rizzulli, Kopp, and all my great friends at NIH Intramural and Extramural. It's a tremendous honor to be here, and I'm glad that you've warned everyone about my fast-talking New York accent. I'm going to talk about a topic that is strikingly controversial to everyone in the general population, but has never been to me. And it's the concept that there are differential distribution of risk variants in certain population groups that put them at unique susceptibility or protection from complex disease. And it is striking to me how many people find that concept threatening or connoting bias. And I think you have to be careful using the data. But basing things on scientific results is clearly where this is going. And I think ApoL1 is an extreme example. But I'm going to take you through many common complex diseases and show you that there are ethnic-specific risk variants in those diseases. Now, it's striking to me as a clinician, as an MD, that when we talk about biologic parameters like kidney function and glomerular filtration rate, nobody would ever argue that there are differences between African Americans and European Americans. As a matter of fact, when we use our equations to calculate kidney function, if we have a 50-year-old male with a serum creatinine concentration of 1, our equations have to adjust for race because creatinine is produced by muscle and there are differences in muscle mass. And if it's a European American who's 52 like me, your GFR is going to be 78. That same serum creatinine concentration in an African American is 95 ml per minute, 17 ml per minute difference. So this is a biologic aspect of a parameter we use to measure kidney function where nobody would argue that population-specific differences exist. Well, the topics I want to cover today, as an introduction, I want to talk about African risk to poor response to treatment for hepatitis C. and African American protection from IgA nephropathy, which is another one of the most common kidney diseases affecting world populations. And these are not areas that we have worked on. And then I'm going to move into the fields we have worked on, including type 2 diabetes, non-diabetic nephropathy, atherosclerosis, and links between bone and blood vessel disease. So just as my couple of introductory slides, I would never want to start a talk like this, and I'm going to speak a lot about genetic factors and genomic factors. Well, let me just tell you, I'm a big believer in the environment and how we have to account for the environment, and I don't want anyone to leave today's talk thinking everything, the basis of disease in all peoples is genes. But the fact is that we are all far more alike than different, no matter where we're from. I would just point out that in addition to the environmental effects of growing up in a separated population group, this less than 1% genetic variation can contribute profoundly to common complex disease. And we all talk about different diets, lifestyles, attitudes, exposures, stressors, and perceived stresses. This is critically important, access to health care. I'm going to show you data that in the general African American population, there's a far higher risk for cardiovascular disease and myocardial infarction than European Americans. However, when you equalize access to health care, the biologic effects come out where African Americans actually have a 50% lower risk of myocardial infarction. So I want to point out these are critical. But as we're learning with ApoL1 and other important risk alleles, that gene-environment interactions may be critical, and they may differ based on population groups because the genetic profiles may be different and the environmental exposures may be different. And this may also contribute to population-specific differences in risk. And the environmental contributors there hold out great hope for cures for genetically based diseases, and that's one of the things we're focusing on. So this is a topic that is exploding in recent years. This is data on treatment response to hepatitis C, pharmacogenomics, if you may. Hepatitis C is 170 million cases worldwide, leading cause of cirrhosis in North America, and we have treatments, pegylated interferon alpha and ribavirin, but these are very toxic and necessarily expensive treatments. The fact is that after treatment, after identical treatment courses, patients of Asian and European ancestry have far higher cure rates, clearing the virus from the circulation than African ancestry populations. And this is a very important paper from 2008 in Nature by David Goldstein's group at Duke, showing that genetic variation in a single gene predicts pretty much half the response to interferon therapy for hepatitis C. And when he looked at his data, whether we were talking about European American populations, African American, or Hispanic populations, you can see sustained virologic response is far higher in individuals who are homozygous for the C allele at this SNP. And there's a graded effect with the poorest response in people homozygous for the T allele. So a lovely dose response based on your genetic makeup. So why would African Americans respond 50% less well to treatment compared to Asians and European American populations? And the answer is right here. If you look at that protective C allele that portends a good response to treatment, the frequency in East Asians is over 70%. European Americans and Hispanics is between 50 and 60%. In African Americans, it's in the low 20%. So the distribution of this allele explains a tremendous amount of the variation in treatment response, a pharmacogenomic effect. Well, we've seen the same thing in IgA nephropathy. And I remember, as we focus a lot on diabetic nephropathy, which is a leading cause of end-stage renal disease in the developed world, the Western world, IgA nephropathy is right up there with it on a worldwide basis. And the interesting thing is it's long been perceived that populations of African ancestry are at lower risk for IgA nephropathy. But many of us clinicians thought that was because of a lack of access to health care. They weren't getting to kidney doctors in time. This is a disease that you must make based on a kidney biopsy. Patients present with hematuria, and the hematuria can progress to proteinuria and then development of chronic kidney disease in a subset of patients. Clearly, in Asia, they screen school-age children with urinalyses because this disease is so frequent that they pick up hematuria early and they follow those patients aggressively and do kidney biopsies in those with progressive disease. But it's been thought that European Americans are in the middle and Africans are at low risk, although again, we wondered if this was because of a lack of biopsy data and access to care. Well, Ali Gharavi's group in New York has just published a paper in PLOS Genetics last year looking at variation in 7 susceptibility loci for IgA nephropathy across the world, and it's shown that this variation explains about 5% of the risk for development of IgA nephropathy. But this is a standardized plot, and you can see distributions of the number of risk variants in these 7 genes associated with IgA nephropathy. So for European ancestry populations in yellow, you kind of see a nice bell-shaped curve right around the midpoint. If you look at Asian populations, you see in red, it's skewed to the right with excess numbers of risk variants. And if you look at African ancestry populations, it's skewed way to the left with a low frequency of risk variants. And if we look at a heat map, you can see all across Asia the highest prevalence of disease and the highest number of risk variants, with a graded effect throughout Asia, Europe, and into Africa, where the frequency of disease is far lower and African Americans are far less frequently affected. So interestingly, it's easier, in my mind, to pick up an African ancestry protective effect than risk effect, because the environment complicates risk. Poor access to care, low socioeconomic status are associated with risk, as are risk variants. But here's an example of a low frequency of risk variants in African ancestry populations. So these 2 examples of hepatitis C and IgA nephropathy prove the point. And I want you to be aware of what a tremendous shift in the paradigm this is. So this is a New England Journal of Medicine sounding board from 2003, not 10 years ago. And I remember reading this paper on a plane where it said minority groups, particularly blacks in the United States, are assumed to be genetically predisposed to virtually all common chronic diseases. Genes are regularly proposed as the cause without genetic data, and the social and biologic factors are hopelessly confounded. So that was a little depressing. But it went on to say that coincidence is not an explanation. The correlation between unsupported genetic inference and social standing of a group is glaring evidence of bias and demonstrates how race can be used to categorize and rank subpopulations. Well, I found this appalling and shocking, and I was very upset by this, and I said, who's doing this stuff? And I went and rushed to the reference section of that list, and that first reference 17, was our work. So I want to make the point to you that in less than a decade, the field has flipped, and it's now appreciated that common risk variants across population groups can associate with disease. So let's start off just quickly with type 2 diabetes, and then I'm going to work my way to the fascinating relationships between atherosclerosis and bone disease. We work predominantly in diabetic populations, as I'll show you, and I'm going to end up with non-diabetic kidney disease. So this is data on the prevalence of type 2 diabetes from the ADA website, very recent data. And you can see that among adults between 2007 and 2009, about 7% of non-Hispanic whites are felt to have diabetes, 12.6% of non-Hispanic blacks, and 11.8% of Hispanics. So clearly a high risk for type 2 diabetes in the African ancestry population. This is data that Don Bowden and I just submitted to the 2013 ADA meeting, so this data is not yet published. But what we simply did was we took the risk allele variants in 17 type 2 diabetes associated SNPs from the major GWAS analyses. So of the 50 or 60 replicated genes for type 2 diabetes in GWAS, we took the 17 most replicated, most powerful, and looked at risk variant frequencies, the risk allele loads, in about 2,000 European Americans, about 800 with diabetes and 1,000 without, and in 4,000 African Americans, 2,600 with diabetes and 1,400 without. And by the way, all these individuals were recruited under the same protocols in the same geographic region of the country over the same time periods. They all came through my office. So there's no difference in any type of recruitment pattern. And to make you comfortable with the genes we looked at, that they're really diabetes genes, PPAR gamma, JAZF1, Tcf7l2, which is the most reproducibly and powerfully associated diabetes gene, KCNQ1, KCNJ11, FTO, the obesity gene, HNF1 beta. I don't think there's much argument in the diabetes genetics literature that these genes and pathways play an important role in susceptibility. So we simply took these 17 genes, which can give you a maximum of 34 risk variants, one from each parent, And we plotted out in African Americans with diabetes versus European Americans with diabetes, what are the risk variant loads? And I think you can see a significant p-value of 10 to the minus 123. And here's the curve in African Americans with a higher risk variant load. Now, look very closely, because I'm going to change the slide. It's going to look a lot similar, though. This is actually the risk allele load in the non-diabetic controls that are African American and European American. So these people don't have diabetes. And let me go back and remind you that in African Americans, the risk variant load is about 22. And in European Americans with diabetes, it's about 18. And when we jump to the non-diabetic group, it moves to the left. It moves down, as you'd expect. But the point is, there's still an increased risk load in non-diabetic African Americans compared to non-diabetic European Americans. And if we put this all together, in all the cases and controls together, we get a p-value of 10 to the minus 267. And I don't think anybody would argue that there are more risk variants in general in these 17 common variants identified by GWAS in African ancestry populations. So individuals with type 2 diabetes carry a higher genetic allele risk load, whether they're African American or European American. But African Americans, regardless of diabetes status, have a higher overall genetic risk, which may contribute to the higher prevalence of type 2 diabetes when combined with a complementary environmental stimuli that would trigger this: obesity, inactivity, dietary excess. So we think there's a genetic background. Just like in IgA nephropathy, there may be a shift of risk variance to favor development of type 2 diabetes in African ancestry populations. But that's not really where I've spent the bulk of my time. I'm a nephrologist, and nephrologists are not only interested in kidneys, but blood vessels and coronary arteries and bone disease. All these things affect our CKD and dialysis population. So we've developed a series of studies, first started by Don Bowden, and many of them are now jointly between Don Bowden and myself, the Diabetes Heart Study family of studies, where we've done some of the most intensive phenotyping in subjects with type 2 diabetes that I'm aware of anywhere. And just to give you a little overview of the samples that we're working with in some of these studies, the original Diabetes Heart Study had 1,400 subjects. Initially, they were all SIB pairs because we were in a linkage era. We were looking for SIBs with type 2 diabetes. And later on in the study, we began to recruit singletons, unrelateds. But we wanted to look at subclinical coronary artery disease using helical CAT scans. And when Don Bowden and I talked, he said, well, African Americans have a lot more cardiovascular disease. We should focus on African Americans. And I said, now wait. As a clinician, I think European ancestry populations have actually more coronary disease. And after a lot of arm wrestling, we agreed to focus on European Americans and have a 15% component of African Americans just to assess if there might be differences. Well, the differences in subclinical coronary artery disease were so profound between these groups, as I'll show you, that led to the African American Diabetes Heart Study, where another 566 African Americans with type 2 diabetes were recruited. With the 15% from here, we have about 750 African Americans, 1,200 European Americans. All these subjects have CT measures of coronary artery calcified plaque, carotid artery calcified plaque, aorta calcified plaque, bone density in the thoracic and lumbar vertebrae, fat adipose tissue volumes around the heart, pericardial adipose, subcutaneous, intermuscular, visceral, hepatic steatosis, kidney function, and a whole host of inflammatory parameters and markers. So this is an extraordinarily well phenotyped study to which we were then fortunate enough to be able to do cerebral MRIs and cognitive function testing to link coronary disease and cerebral microvascular disease for blood vessels throughout the body. And we've recently started the African American Diabetes Heart Study longitudinal, where we're bringing back 300 of these people to look at coronary artery calcium progression. And I'm going to show you some of the fascinating relationships between vitamin D, bone disease, and calcified plaque. So this is the framework that we work in the cardiovascular realm, separate from nephropathy. And I want to preface this by saying higher rates of myocardial infarction and cardiovascular disease are clearly seen in African Americans relative to European Americans, and a lot of this reflects, unfortunately, access to health care and socioeconomic status. There's no argument about that. But I don't know if you're aware that when you look at populations like the Veteran Administration population, Kaiser Permanente HMO, and the Medicare or CMS ESRD program, the dialysis programs, every time you normalize care and equalize access to care, significantly lower rates of myocardial infarction are observed in African Americans relative to European Americans. The differences in these two studies are 50% lower risk of MI. Actually, in the dialysis population, it's striking that African Americans see a nephrologist later in their course. We think that they've had disparities in care for their whole lives. But if you just reverse these things for a few years, you start to see the biology come through. So this is a fascinating paper in the VA population by Dr. Young from 2003. And this study had 430,000 subjects with type 2 diabetes. And I want you to see here, they make the point that all minority populations, African Americans, Asian Americans, Native Americans, had lower risk of cardiovascular disease compared to European Americans. And this is a major table from the paper. You see the Caucasian group along the top is normalized to a risk of 1. And when we're looking for kidney disease, end-stage renal disease, or diabetic nephropathy, African Americans, in the 2 or 3 year period they were followed in this study, had a significantly higher risk of kidney involvement than European Americans, just as we expect, just as we see in the USRDS. But if we look at the risk of cardiovascular disease or myocardial infarction, there's an odds ratio of 0.51, half the risk in African Americans compared to European Americans, at the same time as we see the excess rates of kidney disease. And this is true of all the minority populations, lower cardiovascular rates when care is equalized. If you don't like the VA study and you want to look at Kaiser Permanente, this is another study that had a total of 62,000 patients, came to exactly the same conclusion. 50% lower risk of heart attack in this HMO in African Americans compared to European Americans. And the survival on dialysis is strikingly higher in African Americans than European Americans. And believe me, it's due to reductions in cardiovascular disease. So what we've been measuring in these diabetes heart study family of studies is a helical CAT scan with no contrast. And this white line that you see here is actually calcium deposition in the left main and circumflex coronary arteries. Now, the presence and severity of coronary artery calcified plaque is a strong predictor of cardiovascular disease events and death, no matter what your population group, no matter what your ethnicity. If you have coronary calcium, you're at a higher risk for events. And we've actually shown that if you add this data to Framingham risk score, it improves predictability of outcomes and may better stratify who needs more intensive therapy. So this clearly reflects atherosclerosis. And in the diabetes heart study with the European American patients, I have to tell you that every week I was getting 2 or 3 panic scores for coronary artery calcium levels, 1,000, 10,000. We've seen 25,000 in some of our patients who were asymptomatic, by the way, and just coming in and getting a CAT scan. And as I do this study in the African American diabetes heart study, I very rarely get a call that it's over 1,000. The differences are striking. So when we compare the 2,000 subjects in the diabetes heart study family of studies, I'll just tell you that our African American population have higher fasting blood sugars and a higher hemoglobin A1C. These are all diabetic subjects. They have higher blood pressures, more albumin in the urine, which is a major cardiovascular risk factor, higher LDL cholesterol, and a higher percentage of current and former smokers, all of which should increase your risk, conventional risk factors for heart disease. Now, you know African Americans also have lower triglycerides and higher HDL cholesterol, which will be protective. But the question is, how much do all these factors feed in? What are the coronary artery calcium scores in these populations? And the differences are simply amazing. In the European American men, the median coronary artery calcium was 1,300. In the African American men, the median score was 166. 8 times lower, in spite of all these excess conventional risk factors. Among women, it's less pronounced. It's about 2.5-fold lower. And the difference were so staggering that this is what led to performance of the African American diabetes heart study to explain this relative protection from calcified plaque that does translate to reductions in events when you have equal access to health care. What I want to talk about now is the tool that we and others have used. And frankly, I have to credit many folks here at NIH, Michael Smith, Jeffrey Kopp, Sherry Winkler, George Nelson, the groups we've been working with. we've applied Regional Admixture Mapping, which is a genetic mapping tool for gene discovery in admixed populations. African Americans are an admixed population with approximately 80% African ancestry and 20% European ancestry on average across the United States. And if there are disease-causing variants that are present more frequently in an at-risk population group, either the group of European ancestry or African ancestry, and we're looking for regions of the genome enriched for the ancestral populations that carry excess risk of disease. This can be done at far less expense than a traditional GWAS. 1,500 carefully selected markers that have markedly different allele frequencies between population groups can screen the genome. And frankly, this is what led to identification of APOL1. And if the risk disease is European in ancestry, say atherosclerosis or calcified plaque, you'd expect African Americans with high levels of coronary artery calcium to have excess European ancestry in certain regions of the genome carrying the risk alleles. If the disease is nephropathy of African ancestry, because African Americans have far more nephropathy than Caucasians, you'd expect excess African ancestry in that region. And this is a figure that I stole from Mike Smith's review paper. If we have population A, let's just say that this is a European population, and B, an African population, with early admixture and recombination, large chunks of the genome recombine, but over multiple populations, the recombination regions become smaller and smaller, and we can tag them with variants that have tremendously different allele frequencies across population groups. When we compare a case group with disease versus a control group without disease at the same marker, we're looking for differences in ancestral population risk at that variant that would go with disease. So if the dark blue here is African ancestry and the dotted line is if the dark blue line is cases and the dotted line is controls, and we see a region with excess African ancestry, that chromosomal region would likely be associated with a kidney disease variant if end-stage renal disease is the phenotype. If it goes the other way and there's a reduction in African ancestry or excess of European ancestry, that would be a European-derived risk variant. So the premise is very simple. You only need 1,500 markers to scan the genome. And this is a paper that our group just published in the last few weeks, where we performed admixture mapping for coronary artery calcified plaque in African Americans from the African American Diabetes Heart Study, and we also included subjects, African American subjects with diabetes from MESA and FHS. So we actually ended up with 1,040 cases. About 700 of them were from the African American Diabetes Heart Study. The MESA study had already published that in MESA, African Americans with higher levels of calcified plaque had higher levels of overall European ancestry, which fit exactly with what we saw on the helical CT scans and the data from the VA and Kaiser and the dialysis registries. So we took this another step and looked for where in the genome was this European ancestry, and the results are fairly striking. So overall, in these 1,040 subjects, African ancestry was 80%. In the group that had presence of coronary artery calcified plaque, so greater than 10 is considered present, you can see there was 21% European ancestry. In the group with no coronary artery calcium, 19% European ancestry, and that difference held after you adjusted for age, gender, the different studies, whether we looked at presence or absence of coronary calcium, or if we looked at it as a quantitative trait. But we actually identified 11 regions in the genome, 3 of which were genome-wide significant by admixture mapping for coronary artery calcium, 8 others that were strongly suggestive with a LOD score of greater than 2.5. 11 out of 11 regions demonstrated excessive European ancestry. There wasn't one single region associated with coronary artery calcium in African Americans of African ancestry. So this is the LOD scores for these 11 regions, and you can see 3.7, 3.0, and 3.4. The top LOD score on chromosome 1 is in the GLIS1 gene, and I'm going to talk to you a little bit about the relationships between bone and vascular calcium. Coronary calcium comes from osteoblastic transformation of blood vessel cells. And actually, GLIS1 regulates BMP2 and BMP7, the bone morphogenetic protein genes. So we're very excited about these regions. But the point I want to make is every single one of these regions there's a higher frequency of risk allele variance in the group with coronary artery calcium that's present versus absent. It's staggering, 11 for 11. So I think admixture mapping is a very exciting tool for this trait, and we're now fine mapping these regions and trying to hone in on risk variance, and these are broad linkage type peaks, so we have to fine map and get down to exactly where the causative alleles are. But there's another example of a higher risk variant burden from European ancestry in the African American population that contributes to coronary disease. And I should mention that the chromosome 9 peak, which we picked up on MALD mapping, is the same 9p peak that's associated with coronary artery disease in GWASs of European populations. This region is not only associated with MI, but also coronary artery calcium in European populations, and now we can show it's also playing a role in African ancestry populations. So a very exciting tool, and we hope to be able to hone down the ethnic differences and susceptibility with those data. Now I want to talk to you about a very hot topic in the world, vitamin D, osteoporosis, bone disease, and associated cardiovascular risk. So we all need to be aware that there's an opposing relationship between bone mineral density and calcified atherosclerotic plaque, or subclinical atherosclerosis. Individuals with low bone density or osteoporosis have markedly higher levels of coronary artery calcium. It doesn't matter if you're black, if you're white. It doesn't matter if it's done in a longitudinal study or a cross-sectional study. There's no paper that has been published that disputes this opposing relationship. And again, there's some physiology behind it. The vascular smooth muscle cells take on a cell program like an osteoblast in bone. So do ethnic specific relationships exist between bone and vascular health? And as a nephrologist, I'm quite interested in this. We love all things mineral and electrolyte and bone. But one of the smartest things that was done in the diabetes heart studies is by Dr. Jeffrey Carr, a radiologist at Wake Forest, is he put this phantom under all the CT scans. So while we're scanning the heart for coronary calcium or the aorta looking for aortic calcium, we have a phantom that we can measure thoracic and lumbar bone mineral density on the helical CT. So suddenly we were measuring simultaneous atherosclerosis and bone density. And I'm perplexed as to why other studies looking at coronary calcium have not looked at bone density. We also look at fat volumes on these scans as well. And the point that I want to make is that the vitamin D axis bone health is tremendously different based on ethnicity. So for example, relative to European Americans, African Americans have far lower 25-hydroxyvitamin D levels. The question is it a problem? I don't think it's a problem. I think it's like GFR. It's a totally different scale in African ancestry populations. And it's not just from having darker skin with lesser activation of vitamin D in the skin. African Americans have higher active 125 dihydroxyvitamin D levels than European populations. They have lower rates of osteoporosis, and that's despite the fact that they have less dietary calcium ingestion and these tremendously lower vitamin D levels. So if you have a low vitamin D level in an African American, why are we supplementing it if their bones are healthy? That's the treatment of vitamin D, is to keep your bones healthy. If there's no bone disease, don't treat a number. The range is physiologically different. We already talked about the fact that coronary artery calcium and vascular calcium is much less in Africans than Europeans, but there's increased renal tubular calcium reabsorption, and as a result, far less calcium is in the urine of African ancestry populations, and they have fewer calcium-containing kidney stones. African Americans have higher levels of intact parathyroid hormone, a hormone that breaks down bone to release calcium and phosphorus, except their bones are resistant to the PTH and they don't break down their bones. So everything that regulates vitamin D, calcium, bone metabolism is on a tremendously different scale in African and European ancestry populations. And I think many nephrologists and endocrinologists fail to appreciate this and think about one normal range for vitamin D. Well, in our African American diabetes heart study, we found some shocking results. Number one, when we look at 25-hydroxyvitamin D levels, we see that, well, actually, I should say, we have not looked at vitamin D in the whites, but there's a perception that vitamin D would protect from atherosclerosis. And I'm going to tell you there's very little data to support this, but there's a feeling in the general community that led to the Institute of Medicine report last year saying there's no data for atherosclerosis or cancer protection from giving vitamin D. You give vitamin D for bone disease. But there was no concern that vitamin D was bad for blood vessels. In the African American Diabetes Heart Study, we saw positive relationships between 25 vitamin D and coronary artery calcium. There were 500 African Americans we studied. I think less than 10 were on supplemental vitamin D. This was just their baseline circulating vitamin D levels. The higher the vitamin D level, the higher the coronary calcium. So we caution general internists and family doctors should not be supplementing vitamin D without bone disease because you might be worsening atherosclerosis. Even more strikingly, it's known in European populations, high vitamin D levels associate with higher bone density and protection from atherosclerosis. In the African American Diabetes Heart Study, the higher the vitamin D levels, the lower the bone density. People thought we were crazy until the Women's Health Initiative published data that in white women supplementing vitamin D, following them longitudinally, hip fractures went down. In African American women supplementing vitamin D, getting it to higher levels, hip fractures went up. So there's an ethnic difference in response to vitamin D and bone health, and because of that inverse relationship, we worry about regions in the genome that may regulate coincidentally bone health and coronary disease risk. GLIS1, is a gene that's holding up so far in the fine mapping and could regulate BMP2 and be a great link. So these are very common diseases: osteoporosis, atherosclerosis, type 2 diabetes, and I think we have to look at population-specific risk differently in African ancestry populations. But I want to end in what I should be talking about as a nephrologist, and this is an African American patient on dialysis, and the story is fascinating. You're all aware of this data, that in the USRDS database, the population risk for end-stage renal disease is nearly 4-fold higher in African American populations compared to European ancestry populations. I will say European populations have the lowest risk of everybody. Native Americans, Asian Americans, Hispanic Americans, and African Americans all have more risk for kidney disease. And when we look at diseases like HIV nephropathy, the African American, European American risk may be as much as 50-fold higher. So a lot of this has been attributed to the environment and lack of access to care, but we were very early on interested in familial clustering of kidney disease. And for those geneticists in the audience, you'll know this must be X-linked because only the men are affected in this family. And this is from a series of papers we did in African American population in the early 90s. There was this disease called hypertension attributed nephropathy, which we never quite believed was caused by high blood pressure, but it was non-diabetic subjects who generally had low levels of protein in the urine and didn't get a kidney biopsy. Basically, that's what it was. We looked at African Americans with type 2 diabetes and nephropathy, lupus and nephropathy, and HIV nephropathy, and this is in the pre-heart era, so this was essentially untreated HIV. And what was striking was these were patients new to dialysis. 24 to 40% of them reported having other relatives who were on dialysis at some point in their life, first or second degree relatives. This was an amazing family clustering. And then we went and looked at African Americans with the same degrees of hypertension, diabetes, anti-nuclear antibodies, and HIV infection, who after a prolonged course did not get renal involvement. And we asked about their families. Hypertensive African Americans without renal involvement, only 8% had a relative on dialysis. Diabetes, only 7%. The index case lacked nephropathy. Lupus, these are people with anti-nuclear antibody disease, renal sparing, and HIV after a long period but no renal involvement, only 6%, none, 7% and 8% had a relative with end-stage renal disease. In the current era, 7% of all African Americans will be on end-stage renal disease replacement therapy at some point in their life, 2% of whites. So it's 3.5-fold higher in African Americans. So these groups with the same high blood pressure, diabetes, anti-nuclear antibodies, and HIV infection looked like the background population. But the minute the index case had nephropathy, their families were flush with nephropathy. And by the way, this doesn't count relatives with chronic kidney disease not yet on dialysis. Well, this is from a paper we wrote in 1993. And I think this is the most important thing we've ever found. These were typical run-of-the-mill African American families that had multiple members on dialysis. I know all these families. I know the people in these families. And here's a family that had 4 relatives, 3 brothers and one of their sons, 4 different types of kidney failure. Every family had somebody with kidney failure attributed to high blood pressure, another brother with diabetes, glomerular disease, and type 1 diabetes. there are families here with HIV nephropathy who have HIV-negative siblings and parents who are on dialysis for other causes. This is very common. Many, many African-American families have multiple kidney failure genes, and this led us to multiple members with kidney failure of different types. And we don't see this in European populations, Hispanic populations, Asian populations. Every family clusters one type of kidney disease. And this led us to hypothesize that there was an overarching kidney failure susceptibility gene in these families, and then if you had HIV or hyperglycemia or anti-nuclear antibodies, you would scar your kidneys, and if you lacked the underlying genetic susceptibility, you might not. And you all are familiar with, the GWAS and candidate gene association studies finding very common variants of small effect, and linkage or family-based studies finding rare variants of large effect. And it's been thought that common variants with large effects are rare, and that's, of course, unless there's selection going on. And we think APOL1 is a great example of a risk variants that are selected for protection from trypanosomal infection that cause kidney disease. And this is from Jeffrey Kopp's landmark Nature Genetics paper that we were fortunate enough to work with him on. And using admixture mapping, Jeffrey showed a single peak on chromosome 22 that had 10% excess African ancestry in patients with focal segmental glomerulosclerosis, FSGS, and HIV nephropathy. Now, the contribution we made to this was that we had many patients with this hypertension-attributed nephropathy on dialysis that were not known to have FSGS, that were thought to have high blood pressure-induced kidney failure, and we saw very strong association in this region between APOL1 and MYH9, this whole genomic region. And in further work that's been done by Marty Pollack and Jeffrey and myself, our groups, it's now shown that APOL1 actually is the gene that's responsible. There are 2 coding variants, and this is a fascinating study of trypanosome exposure to serum from African-American individuals based on their APOL1 genotype. And you see this is a wild African-American with no risk variants in APOL1, and that's a very happy, healthy trypanosome swimming around with a normal lysosome. But if you take serum from individuals who have the G1 risk variant or the G2 risk variant, you see lysosomal swelling, because when the APOL1 protein is taken up by the trypanosome, it unfolds, inserts into the lysosomal membrane as a chloride channel. Chloride flows in, lysosomal rupture with death of the parasite and protection. So 1 copy of a G1 or G2 risk variant, protection from trypanosomal infection. 2 copies, 1 from your mother, 1 from your father, tremendously increased risk for kidney disease. This is African sleeping sickness or trypanosomal infection is transmitted by an infected tsetse fly, and there's an example of a canker sore after a sting of an infected tsetse fly with the trypanosome injected into the skin, and eventually it'll make its way into the circulation. Now, there are tremendous clinical applications on the horizon from this major APOL1 discovery, but I'll just tell you that the risk variants that cause kidney disease, the G1 and G2, and we're working hard to find other risk variants, are virtually locked into African ancestry populations, and we think they're a fairly recent origin, the last 10,000 years. Asian populations, European populations virtually lack the G1 and G2 risk variants. They're just not there. So we now know that this kidney disease that's long been attributed to high blood pressure is not. And it's actually a relative of FSGS. It's in the APOL1 focal segmental family, which is a primary kidney disease that leads to secondary hypertension. We know that there's APOL1 expression in podocytes, and currently the field is up in arms about whether it's also, the mRNA is in proximal tubular epithelial cells and endothelial cells and blood vessels. This work is actively ongoing. APOL1 binds to HDL cholesterol, and we don't know if HDL cholesterol is impacted that can cause vascular disease in the kidney, or if APOL1 doesn't bind as well if it's a risk variant, so it falls off HDL and it's filtered at the glomerulus and it's toxic to the tubules where you see tubular scarring. So this is active investigation. But we can already start talking about genetically screening high-risk populations, preferentially African ancestry kidney transplant donors, and I can show you data how we can now take populations and dissect out, just based on the powerful effect of APOL1, for non-diabetic kidney disease. So the first thing this finding did is it reclassified a spectrum of kidney diseases from idiopathic focal segmental glomerulosclerosis, this scar, to collapsing FSGS, as we see typically with HIV infection, but there are other causes, to the disease that's been called focal global glomerulosclerosis, which, based upon ApoL1, this is all a spectrum. Heavy proteinuria, mild to moderate proteinuria, very low proteinuria, yet they're all associated with ApoL1 very impressively. To tell you how impressively, you know, when we talk about genes we find in GWAS, the Tcf7l2 gene is the most powerful type 2 diabetes association gene, most replicated gene. Actually, Nicole Allred in our group found the causative variant in Tcf7l2 because of the smaller haplotype blocks in African American populations. But the odds ratio is 1.4 with a population attributable risk probably less than 5% for diabetes. When we talk about ApoL1, first of all, 12% of African Americans have two risk variants, one from their mother, one from their father. 39% have one risk variant, and about 49% don't have any risk variants. The population attributable risk for FSGS and HIV nephropathy is 70% in a paper from Jeffrey Kopp, and probably well over 50% for hypertension attributed nephropathy. The odds ratios are 29 for HIV nephropathy, 17 for FSGS, and over 7 for this non-diabetic nephropathy. Variation in ApoL1 fully explains the excess risk for non-diabetic end-stage kidney disease in African Americans relative to European Americans. It's not access to care. It's not poor treatment of blood pressure or anything else, which clearly can exist. But it's this genetic variation which explains the vast majority of that excess risk. And we've known that donor kidneys from individuals of African ancestry don't survive as well after kidney transplantation compared to European donated kidneys. ApoL1 seems to fully explain that risk as well. What's striking is that we see much weaker associations with mild nephropathy. It's associated with severe nephropathy. And not everybody, not all the 12% of African Americans with two risk variants will go on to get kidney disease. We think that there are modulators or second hits. So there may be ApoL1 second gene interactions that we're actively looking for. And of course, there are environmental interactions. In HIV infection, in the pre-HAART era, 50% of HIV-infected subjects with two ApoL1 risk variants would get HIV nephropathy. So HIV is clearly a modifiable risk factor. And now that we have heart therapy, this disease is going away. There are other HIV-associated kidney diseases with immune complexes, or cardiovascular disease in long-term survivors. But collapsing FSGS or HIV nephropathy is going away with heart therapy. So modifying the environment can treat a disease that's genetically transmitted. We're now looking at other second hits. We've shown that patients with lupus that progress to dialysis, but not mild nephritis, are ApoL1 associated. And we're looking at other viruses that live in the kidney, much like HIV, that may contribute to protection or excess risk that may be treatment targets. Sickle cell nephropathy or hemoglobin SS is another gene that appears to interact. And we now see a broad spectrum of diseases. And I'm convinced that the reason some people have focal global glomerulosclerosis or focal segmental collapse is different second hits, different environmental factors or second genes that interact to give us a different final phenotype. To wrap up and just show you how clinically useful this is, I want to present a case to you of a 60-year-old African-American woman, diabetic for 10 years, proteinuria 4 grams per day. Strikingly, she didn't have diabetic retinopathy, and she had a brother who was on dialysis who had non-diabetic nephropathy. We genotyped her for ApoL1. She was a 2 risk variant carrier, and my partner wrote on her CMS 2728 form, diabetic nephropathy. And I said, this woman does not have diabetic nephropathy. This woman lacks retinopathy. She has a brother with non-diabetic kidney disease, and she has the ApoL1 risk genotype. She needs a kidney biopsy. Well, we were both right. She had diabetic nephropathy, but she also had collapsing FSGS and focal global glomerulosclerosis, so a primary kidney disease unrelated to diabetes. Why is this important? If the NIDDK wants to do a trial for a treatment for diabetic nephropathy, don't you want patients in the study who have pure diabetic nephropathy? These are patients, African Americans, who have coincident diabetes, because it's very common, proteinuria, which we see with diabetic nephropathy and FSGS, and they don't get kidney biopsies very typically. Matter of fact, most of the time they're not biopsied, and they're given a clinical diagnosis. We can now reach into those cases and genetically dissect cases of non-diabetic nephropathy that wouldn't respond to the study arm treatment and pull them out. So without a kidney biopsy, we can now genetically dissect non-diabetic from diabetic nephropathy. This is a striking paper we published a couple of years ago. These are African-American transplanted kidney from deceased donors. And these are the deceased donors who had 2 ApoL1 risk variants and the allograft survival. These are the African ancestry donated kidneys that had less than 2 ApoL1 risk variants. Now, I have to tell you, 50% graft loss between 2 and 3 years is a bad outcome. But if you look at African ancestry donated kidneys that didn't have 2 ApoL1 risk variants, the survival is identical to European donated kidneys. So it's not that African ancestry kidneys are not going to work as well. It's kidneys with 2 ApoL1 risk variants are not going to work as well. And this has tremendous clinical implications because we can rapidly screen donors. And these outcomes actually are worse than the outcomes of expanded criteria kidney donors. Donors that we take that are less than optimal, but the patients need a transplant so badly, we'll take that kidney. Well, ApoL1 2 risk variant kidneys fare worse, far worse than ECD kidneys. So potential recipients should know. They should be informed to say, do you want this kidney? I think this has a tremendous clinical application. And then the last study I'm going to show you, and I'll wrap up. This is the African-American study of kidney disease and hypertension trial data. You guys are all very familiar with this study. This was performed on the basis that hypertension contributed to development of renal failure in African-Americans without diabetes and low-level proteinuria. And if we can get the blood pressures low enough and use enough ACE inhibitor drugs, we can treat this nephropathy well. Well, as you can see, in the first 3 years of the study, the group that received ACE inhibitors and a low blood pressure goal did a little bit better than the group without ACE inhibitors and a usual blood pressure goal. So at 3 years, these patients were all switched to aggressive therapy. Now, when I look at the natural history here, this looks to me, and this, by the way, the endpoint is death, doubling of serum creatinine, or starting dialysis. And most of the endpoints were renal endpoints, not death. The fact of the matter is, at 10 years, nearly 60% of participants with aggressive therapy and normalization of blood pressure reached a study endpoint. This looks like a primary kidney disease to me. This looks like FSGS or something else we're used to treating. Well, this is a paper that's just been published in the January Kidney International with Jeffrey Kopp and Sherry Winkler, Mike Lipkowitz, and our group, where we looked at ApoL1 associations in ASK participants versus healthy controls from Wake Forest, where I am, because ASK had no healthy controls. If you looked at all the ASK subjects we had DNA available, 663, 600 controls, the odds ratio is 2.5. P-value is 10 to the minus 8th. But if we looked at the people in ASK whose creatinine rose over the course of the study to greater than 3 or who ended up going on dialysis, the odds ratio is 4.6. And if we look at the group who started the study with proteinuria over 600 milligrams a day, which, by the way, you know, nephrotic range proteinuria, diabetic nephropathy type proteinuria is 3.5 grams a day or higher. This is 600 milligrams a day up to 2.5 grams. This is relatively low-level proteinuria. But in those subjects who had proteinuria at baseline, the odds ratio is 6.3. There was only 100 of those subjects and 600 controls. To get an odds ratio of 6.3, this is a powerful effect gene. It's a progression gene. And this is an accompanying editorial by Carl Skorecki. Finally, other people in the world are coming around to the concept that this is hypertension misattributed kidney disease. This is a primary glomerular disease which also has vascular and interstitial scarring in the kidney that's attributable to ApoL1. And the goal that we have been espousing is these variants interact with environmental triggers to cause progressive kidney disease with secondary hypertension. We've got to find the environmental triggers that will lead to the cures. And our new concept about ApoL1 nephropathy is it's not an initiator of kidney disease, but if you have sickle cell disease, if you have HIV infection, anti-nuclear antibodies, or these primary variants where we don't know what the second hits are yet, in the presence of 2 risk variants, your kidney disease will progress. Mild kidney disease in African ancestry, population-based studies, is only weakly associated. It's progression. So to conclude, African ancestry enriches for risk variants for type 2 diabetes, for this non-diabetic family of kidney diseases and for poor response to hepatitis C therapy with interferon alpha. European ancestry enriches for atherosclerosis, MI, and coronary artery calcified plaque. We think likely osteoporosis as well, with a link to vitamin D metabolism. And when we've done admixture mapping for vitamin D levels, we see genome-wide significant differences in African Americans with high versus low levels. And of course, IgA nephropathy associates with Asian and European risk and relative protection in Africans. The problem is that suboptimal healthcare access and environmental effect, can reverse these genetic predictors. So you don't see the European risk for atherosclerosis in a general population because Europeans have better access to healthcare. You need to equalize that. And my final slide. Although 99% or more of the genome is shared and we're all far more alike than different, the remaining less than 1% can produce marked differences in disease susceptibility and treatment. We must consider the effects of ethnicity and ethnic-specific variants and the environment together to understand these complex socio and genetic factors. And I'm always aware that in the old days, when I was proposing human genetic studies in the early 90s, and every grant that was funded was somebody's animal model of hypertensive nephrosclerosis because it better mimicked human disease. We don't even know a good model of human disease because ApoL1 nephropathy doesn't exist in anybody but African ancestry populations. Caucasians, Asians don't get that disease. So we must be very careful what we talk about, and there are ethnic-specific variations in disease risk. So I want to thank you all for this tremendous honor of coming here, and I wanted to thank my colleagues, Don Bowden, Carl Langfeld, and my collaborators at NIH from the ASK study, the FIND study, Drs. Rizzulli and Kimmel, and all the agencies that have funded our work. Thank you.
Terrific. We have time for some questions. There are microphones in the aisles so that people listening on the web can hear the questions while people are thinking about that. Barry, let me ask you, in terms of this controversy about whether it's a good thing or not to point to variations between populations that may play a role in disease risk, I think you'd agree that it provides us with insights, and the ApoL1 is just one example of pathogenesis that we might otherwise not have discovered. But isn't it also appropriate to imagine that ultimately we will want to get away from the use of ethnic labels as sort of sloppy surrogates of what you're really interested in, which is the individual and the individual's genetic and environmental makeup?
Absolutely. So, we look at everybody as a mixed bag of different ancestries, and we can calculate and compute that fairly well. The thing that bothers me is that we use a term like hypertensive kidney disease equally in African American populations and European American populations. When you treat hypertensive CKD, somebody with kidney problems that's white, you can stop the kidney disease. It won't progress. But in African Americans, it does progress, as you saw in ASK. So the labeling of the disease is just as important as the labeling of the population. But of course, we want to get down to the genome level, and we're all different. And finally, the last thing I'll say is, as long as the data is used objectively, I don't like to hear medical students and residents present a case as a 60-year-old female. I want to know if they're African American or European American. It's just a little extra bit of free data that, it may not always track directly with disease, but it's a lot less expensive than a CAT scan or an MRI, which is frequently equivocal. So as long as you don't apply bias, I want to know ethnicity when it can track with disease, but I also want to know the real genetic susceptibility that underlies it.
Yes.
Well, congratulations for an excellent clinical study showing some of the parameters related to coronary artery calcium level as well as kidney disease. So in these days we hear the larger use of vitamin D in a cancer and quite a few other areas that shows beneficial effects. In fact, I increase my vitamin D uptake every day from 600 international unit to almost 1200 now. So you mentioned about some of the variations and how does it affect people like us in terms of this vitamin D effect on coronary calcium level?
Well, so I think the question is, what are our targets as clinical physicians? I don't think we know the normal range of vitamin D in African ancestry populations. I think the key is we want to know the bone phenotype. We want to know if they're osteopenic or osteoporotic, whether they're African American, European American, Asian, doesn't matter. I want to know, do they have bone disease on the DEXA? And if they have bone disease, and of European ancestry, they're going to respond to vitamin D. It makes things better. I don't think we have the answer for African ancestry populations yet, and more work needs to be done. But I would just caution against widespread supplementation without bone disease, because we don't know what it does to coronary artery calcium, and frankly, it worsens bone density, we think.
In your selected gene studies with 17, and I see some of the genes from Notch to quite a few other ones that relates to this transformation from smooth muscle cell to osteoblast-like cells really. So what is the cell into the coronary artery that is responsible for the calcification? And what are the other parameters in the plasma that affects that calcification?
Well, the cells are smooth muscle cells in the media, we think, and they change to a Runx pathway. They get all the signaling of an osteoblast, but we're still in the fine mapping phase. I just wanted to make the point that with ancestry mapping, 11 of 11 regions that had different ethnicity with high coronary calcium in African Americans were of European origin. That was the only point I'm making. We're going to find those genes in the coming months, I hope.
Good luck. We need the study.
Thank you.
Well, that was an excellent presentation. Thank you so much. And in follow-up, we're studying vitamin D levels in African immigrants to the United States. And I'd like to tell you that we just analyzed the data yesterday, and that 60% of African immigrants to the United States have insufficient vitamin D levels according to IOM standards, and 90% have low vitamin D levels according to the Endocrine Society levels. And we are going to relate that to bone disease from DEXA scans. So it'll be very interesting and important, I think.
It's critical to link that to the bone phenotype, so I applaud you, and that'll be very exciting data to see.
Hi, thank you for giving us this excellent talk. I have one follow-up question to what I don't know her name. So with respect to the diaspora, so you're looking at a small subset of Africans worldwide. Have you looked at, say, pure African populations from the continent as well as in the Caribbean?
Yeah,
there's an epigenetic phenomenon that's really probably the most striking phenomenon that you're seeing.
Yeah, that's a great question. We have not done that work, but after my meeting with Dr. Ratimi today, I'm very optimistic that these types of collaborations will develop.
Maybe he'll answer the question, having just come to the microphone.
Barry, again, excellent talk, and we've been discussing this. And also, as full disclosure, Richard Cooper is my mentor. So I trained from under him. But the point I wanted to make also is that, following the comment from Dr. Collins, it's really that, yes, we see these ethnic differences, but it's not a characteristic of that ethnic group. Because as you showed, sometimes it's 12% of that group that carry a particular genetic variant. So when we say African American or African, we distort that distribution that we see. So the fact that something is more frequent doesn't define that population. I think that's where we need to be, to be careful.
And I agree, and I think we're talking about population groups, not really ethnicities and races, and we're all different mixes of our ancestors' variants. I agree fully, and if my talk was taken any other way, I apologize.
In the terms of the second hits that you listed for the APO1 effect, at least two of them, as I look quickly, were protective in malaria. And I'm wondering if that is the hemoglobin S and SLE risk factors are protective in malaria and severe disease. And I'm wondering if what you're thinking is in terms of how these were selected given APO1's protective effect in sleeping sickness. Are they interacting the same way in those infectious diseases initially?
I have to say, we haven't quite figured this out yet. And with anti-nuclear antibodies, I wasn't aware that protected from malarial infection. But the fact is that APOL1 is not associated with mild lupus nephritis. And actually, we looked at this with Jeffrey Kopp and Jim Bailou's samples here at NIH, and we didn't see an association with just mild lupus nephritis that responded to therapy. You need progressors all the way to dialysis. Yeah. So I don't have the answers to how those factors interact yet. But we're most interested in other viruses. Non-HIV viral infections with a renal reservoir seem to us to be very relevant for looking at combined environmental risk and developing treatments to prevent kidney disease.
Okay.
Well, this has been a very interesting presentation, and there will be a reception just now in the library for those of you who would like to continue the conversation, but please let's thank our speaker again.
Thank you.